Hey yo, what's up to everyone and to my homies listening right now? It's Anton talking right here, and together with Vaughn, Broderick, and Matthew, we're gonna talk all about bacteriophages. So you're gonna ask, what are bacteriophages and what the heck are these? So a bacteriophage is a type of virus that infects bacteria. In fact, the word bacteriophage literally means bacteria eater, because bacteriophages destroy their host cells. The bacteriophage also has a structure, which consists of the capsid head, and inside the capsid head contains the nucleic acid or the DNA of the bacteriophage, and while at the tail consists of the collar and the sheath, which also includes the base plate, the spikes, and the tail fiber. So you're also gonna ask, who discovered bacteriophages? So bacteriophages were discovered independently by this English dude named Frederick W. Thwart in Great Britain at the year of 1915 and by Felix de Herelle in France at the year of 1917. But it was Felix de Herelle who coined the term bacteriophage, meaning bacteria eater, to describe the agent's bactericidal ability. So now we're gonna go to the process of bacteriophages. So the first process of the bacteriophages is the lytic cycle. The lytic cycle is named for the process of lysis, which occurs when a virus has infected a cell, replicated new virus particles, and bursts through the cell membrane. This releases the new virions or virus complexes, so they can infect more cells. The lytic cycle involves 5 different stages, which involves the attachment stage, the penetration stage, the biosynthesis stage, the maturation stage, and the lysis stage. All of these stages are going to be continued to be explained about by Vaughn.
So the first stage is the attachment stage. It is the stage which the phage interacts with specific bacterial surface receptors. Most phages have a narrow host range and may infect one species of bacteria or one strain within a species. This unique recognition can be exploited for targeted treatment of bacterial infection by phage therapy or for phage typing to identify unique bacterial subspecies or strains. The second stage of infection is entry or penetration. This occurs through contraction of the tail sheath, which acts like a hypodermic needle to inject the viral genome through the cell wall and membrane. The phage head and remaining components remain outside the bacteria. The third stage of infection is biosynthesis. This applies to new viral components. After entering the host cell, the virus synthesizes virus-encoded endonucleases to degrade the bacterial chromosome. It then hijacks the host cell to replicate, transcribe, and translate the necessary viral components. Polymerase genes are usually expressed early in the cycle, while capsid and tail proteins are expressed later. In the maturation phase, new bacterial phages are created. To liberate free phages, the bacterial cell wall is disrupted by phage proteins such as lysozyme. Then, here comes the final stage, which may be referred as lysis or release. Mature viruses burst out of the host cell in a process called lysis, and the progeny viruses are liberated into the environment to infect new cells. Next up is the lysogenic cycle. In a lysogenic cycle, the phage genome also enters the cell through attachment and penetration. A prime example of a phage with this type of life cycle is the lambda phage. During the lysogenic cycle, instead of killing the host, the phage genome integrates into the bacterial chromosome and becomes part of the host. The integrated phage genome is called the prophage. A bacterial host with a prophage is called a lysogen. The process in which a bacterium is infected by a temperate phage is called lysogeny. It is typical of temperate phages to be latent or inactive within the cell. As the bacterium replicates its chromosome, it also replicates the phage's DNA and passes it on to new daughter cells during reproduction. The presence of the phage may alter the phenotype of the bacterium since it can bring extra genes. As said, there is a change in the phenotype of the bacterium. This change is referred to as lysogenic conversion or phage conversion. Some bacteria, such as Vibrio cholerae, is less virulent in the absence of the prophage. The phage infecting these bacteria carry the toxin genes in their genome and enhance the virulence of the host when the toxin genes are expressed. In the case of V. cholera, phage-encoded toxin can cause severe diarrhea. During lysogeny, the prophage will persist in the host chromosome until induction, which results in the excision of the viral genome from the host chromosome. After induction has occurred, the temperate phage can proceed through a lytic cycle and then undergo lysogeny in the newly infected cell. So, this is the end of the podcast, and thanks for listening or watching.